All right, guys, so we are going to be starting with our sheep brain dissection today. So as you can see, this is a sheep brain. So we currently are looking at the superior surface. This is the anterior aspect, and this is the posterior aspect. Okay, so one thing you notice with a sheep as compared to a human, or hopefully you've noticed, but if not, I'll tell you right now, sheep walk on 4 legs and humans walk on, well, 2 legs, okay? So this changes the orientation of the brain. You can see that their brain is basically one long line. But for us, you guys have been looking at the model, so you already know that our brains are shaped more like this, where we have this section bent down because we walk on 2 legs, okay? So essentially what we're going to do first is we are going to do a view of the external features, then we'll go ahead and dissect our brain, and then we'll look at the internal features, okay? So first off, we have the cortex up here. And the anterior portion of the cortex, of course, is your frontal lobe. Then you're going to have 2 parietal lobes on either side, and a temporal lobe over here, a temporal lobe over here, and your occipital lobes are going to be in the back, okay? So this is all part of your cerebral cortex, okay? So the cerebral cortex, remember, can also be called the cerebrum. It's also important to notice that the cerebral cortex is made of all of these little wrinkles, okay? So a wrinkle is called a gyri, and the space between the wrinkles is called a sulcus, okay? So this entire thing is covered in gyri and sulci, okay? Now, you'll notice that I can't really stick my pointer down into a sulcus, and the reason for that is we still have a little bit of meninges on here, but this is only the pia mater. You can tell because on the back side here, or rather the inferior side, we have a very tough layer here. That is very obvious, and the tough layer would be the dura mater, okay? But the pia mater continues to protect on this side. And I'm going to see, there we go. I broke through, and when I broke through, you can see that there is a little bit there, and you can easily go in once you break through. But that is your pia mater right there, okay? And there we go. So I'm inside the pia mater. You can see it holds itself up even though the probe, I'm not touching it because the probe is attached to the pia mater, okay? So the other important thing here is we have this long thing here. This isn't a sulcus. This is your longitudinal fissure. The longitudinal fissure is separating the right and left halves of the brain. Now, you know from your meninges that there is a extension in the longitudinal fissure, which is the falx cerebri. And there is a little bit of it, on mine at least, in this anterior portion. You can see right in there. And so that's why my probe is kind of under it right now. So my probe can go in here, but it can't go any further because the falx cerebri is there, okay? Now, the other dura mater, or the other, meningeal extension is right here. Now, you can see there's this kind of triangular portion here that was in between the cerebellum here and the cerebrum here. And so that is your tentorium cerebelli. Most of it isn't cut off, but on both sides there's a little tiny triangle of it left that can be seen, okay? So that's your tentorium cerebelli. So, let's see, what's next on your list? We found all that. We've said this is the cerebellum. The vermis is essentially the line in the center of the cerebellum. It looks a little bit different in sheep than it does in us. We also can see our brainstem. So our brainstem is this portion right here. The inferior, well, it's inferior on us, but posterior here. The posterior most portion on the sheep would be the medulla oblongata. And then we have this rounded pons here. And up under here is going to be the midbrain. And once we do some of our dissection, we'll get a better look at the midbrain, okay? Now, as we said, we still have our dura mater attached here. The main reason for that is because if we pull this off, we would lose a lot of these cranial nerves. So we want to make sure we see the cranial nerves before we pull off the dura mater, okay? So first off, we have cranial nerve 1. Cranial nerve 1 is on both sides. Remember, that gives you a smell sense. It's called the olfactory nerve. The tip of it is called the olfactory bulb, and the portion behind it is called the olfactory tract. Okay? And then we get to cranial nerve 2, which is the optic nerve. We have one side here and one side here, and this place where they cross over is called the optic chiasm. Okay, now cranial nerve 3 I think is buried under here. Same thing with cranial nerve 4, but this really big guy here on either side that has been cut off would be cranial nerve 5, the trigeminal nerve. Now how do we know that? Trigeminal nerve is huge, okay? And so this huge guy is on either side, all right? We can also see, or maybe we can't. Yeah, I think we might have to cut away some of this to see many of the others. We do have this little guy over here, and this little guy is probably 9, I would think, but it's hard to tell for sure, okay? So, let me see. Can we get anything else? We got all those. Let me pull this back a little bit. All right, I'm going to get my tweezers, and I'm going to pull this back. And when it's pulled back, we can see that we have another lobe right here, okay? And this is the piriform lobe. The piriform lobe is present in sheep. It is not present in humans, but that is what we're seeing here. And then we have our rhinal sulcus that is going just above the piriform lobe, okay? And I think until we do a dissection, that's the best we can see right now because we just have too much of the meninges in the way. So we'll see in a sec. All right, guys, so we have our scalpel. So we are ready to go ahead and do our cut. So 2 very important things whenever you are doing a dissection. Always, always, always cut away from yourself. You never know when the tissue is going to be a different consistency than you're expecting it to be, and all of a sudden the knife can get away from you. So you always want to cut away from yourself. You also always want to anchor what you're cutting so that it doesn't wobble around. If you have ever tried to cut a grape, let's say, and you wanted to cut a grape in half but you didn't hold it still, wouldn't work very well, okay? It's going to fly all over your plate. So you always anchor your specimen, you cut away from yourself, and I said 2 things, but I'm going to add a third one. Make small, smooth cuts, okay? Don't just go, because then if you do something wrong, you have no way to fix it. But small, smooth cuts. So what we're first going to do is we're going to cut through this portion. Small, smooth cuts, okay? And then we're just going to keep cutting deeper and deeper with our small, smooth cuts, and you can see that I'm putting traction on it as I'm doing that so that it slowly opens up. Small cuts all the way across. And believe me guys, I have done surgery on human beings and I have dissected human cadavers as well. I'm not telling you to do small and slow and cut away from yourselves because you're amateurs. I'm saying because this is the correct way to do it. All right, don't think like, oh, I've done this before, I know what I'm doing. I do too, believe me, okay? But this is how it's supposed to be done, okay? So, essentially, we have just cut into 2 halves. All right, so now that we have our 2 halves here, what we're going to go ahead and do, and we're going to turn it around, and now we're going to cut through our cerebellum and our brainstem. So again, we're doing smooth, short cuts, okay? And we're still cutting away, all right? Cutting away in our smooth, short cuts, and we now have 2 complete halves. So the important thing is you are now completely done. No more cuts will be happening. So put your scalpel away. All right, guys, so we're going to go ahead and put this piece aside, and we are going to focus in on the inner portion here. So we still have our frontal lobe, parietal lobe, occipital lobe here. We also can see that we have our corpus callosum up top here. We have a little extra piece of tissue that we'll just go on ahead and get out there. So that's our corpus callosum at the top. Now, directly underneath that, you see that there's kind of a glistening thing here. This is septum pellucidum. So septum pellucidum, and underneath that is the fornix, okay? So we can also see this area here is the thalamus. You can see the little eyeball of our pregnant seahorse. This is the thalamus, and then the nose portion coming towards the snout would be our hypothalamus. This is our optic chiasm that we have cut open. You can see the rest of cranial nerve 2 over here, okay? Now, just under his snout area, this kind of spot here would be the mammillary bodies. Mammillary bodies is right here, okay? We also have the midbrain here, the pregnant seahorse's belly, the pons here, and our medulla oblongata. This, of course, continues to be the cerebellum, but when it's cut open, we can see this like vine-like stuff, which we call arbor vitae, okay? Now, these 2 structures together are called the tectum. This top one is the superior colliculus, and then this is the inferior colliculus, okay? I shouldn't say they are the tectum, they are found within the tectum, okay? So remember, these 2 here and the 2 from the other half together would be the quadrema. And so they're within the tectum, superior colliculus and inferior colliculus. And that takes us to our lateral ventricle, which is here, septum pellucidum, remember right here. We also have the 4th ventricle here, and we have the cerebral aqueduct here that is connecting those 2. We have our pineal gland just above our superior colliculus, and I'm just double checking that we didn't leave anything out from your list. I think, oh, we did. Cingulate gyrus, okay? Cingulate gyrus is right here. And we got everything else. So that is everything that you need to know for the sheep brain dissection.